Hi everybody, welcome. We got our latest Lion Lunch and Learn. Today we got the coolest group of guests coming to us from the Rokyta Lab at Florida State University. So let's listen up, let's give them a hand of welcome.
All right, so technically I'm in charge, so I'm Dr. Darin Rokyta, professor at FSU. These are my graduate students, Emilie Broussard, Schyler Ellsworth, and Mike Hogan. They actually do all of the work in the lab, and so I'm going to let them present what our lab does, and then we'll take questions. Hopefully, we're good. All right, take it away.
All right, so we're just going to do a brief overview of the science behind what we do in the lab and then some of our individual research projects and what we actually work on in our Venom lab. So to start off with, the underlying concept that underlies most of our research is the central dogma of biology. So is that a term that you guys have heard before? Maybe, maybe not.
We actually haven't gotten to that yet this year.
Okay, cool.
We have in the years past. So.
Perfect. Well, this will be a good little primer for you then. So the central dogma, or the central idea underlying biology, is that you start off with your DNA, so your genetic code, and then that gets transcribed into RNA, which later gets translated into the actual proteins that make up what you can physically see of an organism's phenotype. And so our lab studies pretty much every step of this process by using the system of Venom. There we go. And then the central dogma is a pretty simple idea of looking at this concept in which DNA transcribed into RNA, translated into proteins. But as always in biology, the real world is a little bit more complicated. So every cell in your body contains the exact same genome. So in total, your genetic code is the same in all of your cells. However, not all of your cells do the same thing, right? Your heart cells do different things than your pancreas cells, which do different things than your liver cells. And so the way that your cells differentiate and do that is through gene expression. So at this step here in transcription is where that varies. So your heart cells are going to transcribe different genes into RNA than your liver cells. And then those are going to be translated into your different proteins, which are going to give your cells your different functions. And so expression, along with the actual traits that the expression produces, is going to be acted upon by natural selection. And so this can happen in a variety of different ways. So you have a couple examples here. So first off, we have a peacock. And so this peacock has a large decorative plume that attracts females. And so the males that have these big decorative plumes are going to be able to reproduce more and pass on those genes to their offspring. We have a hummingbird here, and its long beak is able to better reach the nectar that's inside of those long tubular flowers. It's able to eat more, extract more resources, and so it's going to be able to better survive and then reproduce, pass on its genes to the next generation. And then here, if you can see, it's very well camouflaged. We have a little sand mouse. And so they've evolved to be very well camouflaged in with the white sandy beaches that it lives on. A lighter color means that predators can't detect it as much, and so it's going to be able to better survive, reproduce, pass on its genes to the next generation. And so what I'm talking about here, the ability to survive, reproduce, and pass on its genes, that's considered the term fitness. So the ability of an organism to survive and pass on its genes to the next generation. And so, angry little hognose. We use Venom as a model system for studying the selection that happens on expression and on these traits. And so we're able to do this because Venom is a very straightforward pathway where we can actually take individual proteins and toxins that are in the venom, get the protein sequence from it. We can also get the RNA sequences by sequencing the venom gland, so the actual tissue that produces the venom. And we can even break that down further and sequence the genome or genetic sequences of the snake as well. So we can get all three levels from the DNA to the RNA to the proteins. So we can have the actual protein. We can see how much it's being expressed through the amount of RNA that's being produced in the venom gland. And then we can look at the actual genetic sequence and see if there's any changes that are happening between individuals or populations or even species. All right, and so venom, you probably have some understanding of what it is. It is a toxic substance that some animals have that allow them to better capture their prey and also to defend them from predators. So venom is a pretty complex trait. It's made up of a bunch of different proteins and toxins, and interestingly, it has independently evolved over 100 times across the animal tree. So obviously, a lot of what we're going to be talking about today are snakes and invertebrates that have evolved venom, but also there are some marine invertebrates like some octopi and some cone snails that have evolved it as well, in addition to some little shrews in mammals that have also evolved this venom. So none of these animals inherited that venom from a common ancestor. They all independently evolved that venom on their different lineages. So it's pretty cool that they've all converged upon this same trait that is so effective at keeping them alive and allowing them to capture their prey. All right, so what is venom? Venom is a really complex cocktail of proteins and toxins, and there can actually be tens and hundreds of proteins and toxins that compose an individual venom. So here is a graph that is showing all of the individual proteins that we were able to identify from the venom of Crotalus adamantius. So that's our local eastern diamondback rattlesnake. So you can see that we have all kinds of different proteins over here, and then the height of the bars is showing you the abundances. So some proteins are really, really common in the venom. You see them in really high concentrations, and others are not so common. And so this is where gene expression and the amount in which genes are being expressed and generated into RNA and proteins really comes into play, because the composition of venom can differ a lot between populations and different species of snake. So sometimes we'll have, say, a phospholipase A2. That's a type of venom protein. That'll be expressed really, really highly in a coral snake, but if you take an eastern diamondback rattlesnake, it'll be expressed a lot less. All right, so how we actually study venom? Well, in order to study the venom, we first have to be able to get it from the organism. And not die. So I'm going to show you a video of how we extract the venom from a timber rattlesnake, I believe is in the video. And obviously, this is a disclaimer that shouldn't be said, but don't try this at home. Whenever we do this on campus, we go through a lot of safety protocols and we take every possible precaution to make sure that no accidents and no snake bites happen. All right, it's a little blurry. That's a diamondback. One of them is a timber, though. I went through the whole video.
Yeah, so we use these snake tubes as a way to get our fingers away from the snake. And just basically convince it to bite that little cup.
How often can you do that for one snake?
You want around 20 days in between to let the venom get replaced. That's a little more clear. Yeah, and we really don't need a lot. All the analysis that we could do on a little drop of venom from a scorpion or on venom from a rattlesnake. So as long as there's enough there to see and you can get a pure sample, it doesn't really matter how much you get.
Wow.
Yeah, so we have a YouTube channel. I'm the one who edited all the videos because that's kind of a pastime of mine. But we do have some, other venom extractions. If you wanted to go check it out, just search for Rokyta Lab on YouTube. You should be able to find it.
Yep.
Yeah.
They got the link.
All right, and in addition, we don't just study snakes. We also study, there we go, the venom of invertebrates, specifically spiders, scorpions, and centipedes. And so the way that we collect venom from them is a little different. They're a lot smaller than snakes, so it's a little bit more of a refined process. So what we do for all of these guys is we knock them out with CO2, so they go and take a nap. Then we strap them down to this homemade contraption that basically just keeps their body still, make sure that if they do wake up and start moving around, that they're not going to bite anyone. And then we use this pair of electrodes to provide a light electrical stimulation to the muscles around their venom glands. That causes their venom glands to contract and expel venom, which we can then collect using a little spatula. So here is a picture of a scorpion. You can kind of see his little claws poking out from where he's strapped down. Their venom gland is in their tail, so in their telson. So we shock their tail and they're able to expel their venom. For the centipedes, their venom glands are in their forcipules, correct? Yep. So we have them bite down around this spatula, provide this electrostimulation. They expel their venom on here. And then a similar thing with the spiders. Their venom glands are kind of around their mouths. So again, we put the spatula there, provide electrostimulation, and they expel their venom. So I have a short video of that too. So this is of a centipede. I don't know how to make this one full screen.
Yeah, so you can see the centipede is asleep right here, so we're able to pick it up, and it's just kind of floppy there. So then we're going to strap it down onto this tube so that its mouth is facing up. And so you can come over and look at these guys after, but you can see those forcipules. So there are these really, there's these pincers that are very sharp, but they have a hollow groove inside of them with a hole. That then we have them bite around this metal spatula. When we provide this little shock, you can see these little droplets of venom come out onto that metal spatula. And so like Mike and Emily were saying, we don't need a lot of venom to do some of our studies. And that is actually a lot of venom for a centipede. That's from one of our big centipedes, like this guy over here. But we've done centipedes about this big before. And we get enough venom to do our analyses. And so it's a really cool process that we get to do with these guys.
Mm-hmm.
And that silly video got like 29 million views. Wow. Which was really surprising.
Yeah. Was it dirty?
Yeah.
Can you do that with the centipedes you find under rocks in your yard?
Yeah, they're just smaller.
Just smaller.
They're a little more delicate. They don't give as much venom. But yeah, the same process can be done.
All right, so when we have the venom, what we do next is we can take either the venom or a tissue sample from the venom gland or from the animal and then put it through a sequencer. So if we're working with venom, we can identify all of the proteins and toxins that are in that venom. If we're working with tissue, we can either extract the RNA or the actual DNA and sequence that and look at the actual genetic sequences of our venom proteins and our venom genes. All right, so now that we've gone through our general overview, each of us is going to take a couple of minutes and just talk about what our individual projects are in the lab and what we work with. So Schyler, I think you are first.
So I study a little bit of a different part of the venom system. So I want to study what's called venom resistance. And so there are a lot of organisms that eat other venomous organisms. And so because they eat these venomous organisms or they want to defend themselves against them, they've actually become resistant to their venom. And so there's some really funny videos if you get on YouTube that show a honey badger being bitten by a snake. It then rolls over, falls asleep for a few hours, wakes back up, and then goes back to eat the snake.
Oh, the honey badgers are just crazy. Oh, it's got a cobra. Oh, it runs backwards. Now watch this. Look, a snake's up in the tree. Honey badger don't care. At nighttime, the honey badger goes hunting because it's hungry. Look, here comes a fierce battle between a king cobra and a honey badger. I wonder what'll happen. Look at this. There's the honey badger just eating a mouse. And then look, get away from me, says the snake. Get away from me. Honey badger don't care. And the snake comes back and it lashes right at the honey badger. Oh, little does the honey badger know, FYI, it's been stung. It's been bitten by the snake, so while it's eating the snake, ew, that's disgusting. Meanwhile, the poisonous venom is seeping through the honey badger's body, and it passes out. Now, the honey badger is just going to pass out for a few minutes, and then it's going to get right back up and start eating all over again. Look at this. Like nothing happened. The honey badger gets right back up and continues eating the cobra. How disgusting. And of course, what does the honey badger have to eat for the next 3 weeks? Cobra.
And so they have evolved this venom resistance. So in their genome, in that blueprint that Emily was talking about, they have genes that help them to resist these toxins and to be able to survive to eat a snake another day or just to move another day.
And so I wanted to do this experimentally to see how we can, how an organism would be able to evolve this type of resistance in venom. So I used centipede venom. So we did extractions like we saw in that last video. We were able to get little bits of venom from a lot of different individual centipedes. I then pooled that together, and I actually inject that venom into fruit flies. So if you guys have ever seen fruit flies hanging around your trash can at home, a similar fruit fly we use in the lab, and I, underneath a microscope, have these little tiny glass needles that I can inject the fruit flies with. So this is a little bit of scale here. That's the same thumb that you can see here. So very, very small flies that I'm injecting with venom. So I inject them with venom that will kill roughly 80% of those flies. So some of the flies will live, and some of those flies will die. And what decides whether they live or die is the blueprint, or their genome that codes all of those different genes. And so what I do is I take those flies, I'll then inject them with the centipede venom. About 80% of them will die, but that 20% that survives, I will then go ahead and sequence that blueprint. So then I can identify the different genes that are helping them to resist the venom. So I did this for 2 different centipede species. So the one local centipede species that we have over here, I did that. Its name is Scolopendra viridis. It looks like this one up here. I also did it with a bright red centipede species that is also local. So if you've ever flipped over a rock in your yard or in the forest and found a bright red centipede like this, I also did it with that species here. Like Emilie was saying, there's different species of venomous organisms have different venom components. So this red species here, we can see has these yellow bars here. So those yellow bars mean that it has a different venom component than this one over here. So I wanted to do this with 2 different venoms that had 2 different functions. So this function of the red centipede here, it destroys tissue. So when it gets injected into an animal, whether that's a human trying to bug it or whether that's a fruit fly by me, it destroys their tissue, and so it'll make it start to decay. This other centipede species mainly has a venom that is neurotoxic. So that means it makes the nerve shut down, or it activates your pain receptors. So if you got bit by that one, it would feel really painful. But if something a lot smaller like a cricket or, again, a fruit fly gets bit by that, it will actually shut down the whole nervous system and make it so their muscles stop working and even their heart stops working. And so 2 very different types of venom I was able to inject into these fruit flies. And then on these 2 graphs, we have the entire blueprint, or that entire genome that is with those different or within that fruit fly. And then above this dotted line on each side are areas of the genome that are helping them to have this resistance to the venom. And so we can see a big portion right here that is resistant to the venom, and that lines up pretty well with this part down here. And so through this experiment, I was able to see that there are certain genes that are helping to cause that venom resistance, but they're also different between the male flies and the female flies. So there's something that makes the female flies a little bit more resistant. However, some of those genes are overlapping between these 2 data sets, and some are very distinct between them. And so the part of my experiment that I'm doing now is I'm actually taking these fruit flies that have some of these parts of their blueprint erased, so they're gone, and then I'm injecting them with venom to see if they actually are helping to cause this venom resistance. And so again, this is one aspect of what's happening with our projects at FSU. And I think Emilie's going to come back and talk to you about PLA2.
All right, and then I have 2 projects that I'm going to tell you about. First up is PLA2 Evolution across rattlesnakes. So for this project, I focused in on just one small part of the entire rattlesnake venom, the phospholipase A2 venom proteins. And then I wanted to see what that gene family evolution looked like across North American rattlesnakes. So I took the transcriptomes, or all of the RNA that is produced in the venom gland, of almost every single North American rattlesnake. It's about 42 species. And I looked for these PLA2 sequences in them to try and figure out which ones are being expressed and where. And so I was able to retrieve all of those sequences and then build this phylogeny. So this is basically a family tree showing the relatedness of all of the different sequences across all of the different rattlesnakes. And you can see that it falls into 3 main groups, which are labeled here in the red and blue and the purple. So there's this basic subunit, there's an acidic subunit, and then there's also this small subunit down here called the Mojave toxin subunit. And when this subunit is present in the snake's venom, it causes them to have really neurotoxic venom. So that venom that Schyler was talking about that attacks the nervous system, not the localized tissue, and can cause a lot of systemic damage across your body, not just at the location of your snake bite. I also was able to compare all of these sequences to try and figure out where exactly natural selection was acting on the individual genetic sequence basis. So I laid out all of the genetic sequences, compared them, and was able to identify individual sites where selection was acting differently in these different lineages. And then the second project I'm going to tell you about is related a little bit to what Schyler does. So Schyler's working on Venom Resistance, and Venom Resistance is a trait that evolves in response to venom. And venom, in turn, evolves in response to venom resistance. And so since we have this relationship happening between these 2 different traits, where when one evolves to become, say, more venom resistant, so if you have a population of squirrels that becomes really, really resistant to the venom of a predator, well then this predator needs to catch up, right? Because it's no longer able to kill as many of its prey as it needs to be. And so it's going to evolve stronger venom in response to it. So this is called coevolution, and this specific relationship is often referred to as a coevolutionary arms race, because we have these 2 traits that are antagonistically interacting, evolving in response to each other, and trying to escape each other. And so I'm really interested in this relationship, and this is a relationship that we've observed in many different species. So for example, here we have the Pacific rattlesnake and its prey, the California ground squirrels. So we've known for a while that these species are in a coevolutionary arms race where the venom in the rattlesnake is co-evolving with the venom resistance in the ground squirrel population. So we're able to see this, we're able to test this. However, we don't know a lot about the genetics that actually underlie this relationship and this co-evolving system. So I wanted to understand a little bit more about what is happening here. And so I'm going to do an experiment where I'm going to use a model prey population. So I'm going to use fruit flies instead of a more natural prey like a squirrel. It would be pretty hard to do an experiment with this many squirrels, and also very hard to get permission to do this experiment with a squirrel. So instead, I'm doing it with fruit flies, which are a lot easier to use and don't have as many restrictions associated with them. So what I'm going to do is take the venom from 3 different species in 3 different lineages that have all independently evolved venom. So I'm going to take venom from a coral snake, a scorpion, just like we have over there, and then also a centipede species, and then dilute those down to 3 different doses. So for each one, I'm going to have a high dose, a middle dose, and then a low dose, which are going to kill approximately 80, 50, and maybe 30% of the population each. And then I'll be able to compare the genetics of the animals that survived versus the ones that died in order to identify the number and identity of genes that are involved in resistance. And so my hypothesis here is that when we are injecting flies at a very low dose, the number of genes that are going to be involved in resistance to the venom and survival of the individual is going to be pretty high. It's going to be pretty complex. Because if you're only injected with a little bit of venom, a lot of factors can contribute to your survival, right? If you are a larger individual or if you have a higher metabolism, that increases your chance of survival because you're better able to handle that venom and clear it from your system. However, at a really, really high dose, all of those other factors don't matter as much. You've just been injected with so much venom that unless you have some sort of super rare, super useful mutation, which is going to allow you to directly counteract the effects of that venom, you're going to die anyway. So at this high dose, I predict that the genetics underlying the resistance should be relatively simple. We should see only 1 or a few genes that are actually going to contribute to this resistance and this survival. So this is a project that is currently ongoing, so I don't have any cool results to show you yet, but hopefully I will soon. And I will pass it over to Mike.
Okay, so yeah, I joined Darin's lab because I thought Venom was like the coolest system for all the reasons you just saw, and then I decided I'm gonna scrap Venom and look at a completely different system. I wanted to see how the genes that control rattlesnakes' ability to find their prey are also co-evolving, again, driven by the same, the same place, their actual prey. So in the same way that Venom evolves under all these different hypotheses, we also expect the traits that enable the snake to find its prey in the first place to be under a similar natural selection. So adaptive traits experience selection towards functional optimums. It doesn't matter if that's a protein in venom or a receptor that allows the snake to find its prey. And again, just like the coevolutionary arms race that Emilie pointed out, we do expect the predator avoidance and prey to be what's driving this. And I like to illustrate this by saying if you can't find it, you can't kill it, and you can't eat it, since all of those have the same outcome of a hungry snake. Relax. So yeah, I've been looking a lot at our Eastern Diamondback genome. We sequenced a full Eastern Diamondback rattlesnake genome and got basically all of the genes, that we were able to identify from it. And I just, I wanted to see how similar is it to a human, and just kind of look at all the genes in the genome. And obviously, the human doesn't have venom, so that's something that's very different. And just to clarify, we didn't sequence Darin's genome. I just used his picture. Not yet. Yeah, maybe we'll get funding to do that. But I thought it was interesting because the rattlesnake genome actually had a similar number of transcription factors as the human genome. So that seems to be kind of a conserved thing in vertebrates. Have any of you guys heard of a transcription factor? Okay, maybe one or two. So yeah, it's okay. I'm not going to talk about that too much, but it's basically a protein that affects transcription, and it's involved in what we call regulatory mechanisms. So, what are these bigger gene networks and how are they regulated? So, again, whether you're looking at venom or you're looking at, a receptor, the bottom line is we're going to be doing some sequencing and looking at the genetics. And I know a lot of you, it's like, yeah, they say they're looking at DNA, but what does that actually look like? What are we actually measuring? If I gave you guys a sequence and I was like, okay, I just, spent thousands of dollars and got this sequence, do something with that. It's kind of hard to visualize what do you actually do? What do you look for? What are the differences? So I'm going to take you through a really simple example and just kind of show you one of my preferred methods to look at variation. And it's, using a simple idea, which is every 3 nucleotides codes for 1 amino acid. Have you guys heard that before? Okay. So here, I have 2 sequences, and again, the nucleotides are up top, and underneath each one is the translated amino acid sequence. So there's 2 different types of mutations that you can have on the nucleotides. One of them changes the translated amino acid sequence. So you can see here we mutate from a T to an A, changes the amino acid sequence from an L to an H. And we call that a non-synonymous mutation. It's non-synonymous because it changes. The other type, over here, we mutate from a G to a T, and that does not change the amino acid sequence. So we call this a synonymous mutation. Where having a mutation there doesn't really change what the actual protein looks like. Now we can basically just count up all of these mutations and classify them as synonymous or non-synonymous, and we can start to make predictions about the direction that selection is going. So if we see more of these mutations that are changing the actual amino acid sequence, we call this positive selection, and that seems, or that represents selection favoring changing. It's changing the protein. We're seeing differences in the genome at a higher proportion than the non-synonymous. Whereas if we flip it around and we see that these sorts of mutations seem to occur at a higher frequency, we call that negative selection, where actually that protein is really, really important. So maybe it's like an opsin in your eye or something really important for a metabolic pathway where if you change that protein, it's going to totally screw up your biology. So we call that negative selection or purifying selection. And so now, when we have a site-by-site, way to analyze these sequences, we can start to do some cool stuff. And so this is a phylogeny or a tree of all the olfactory receptors in the eastern diamondback rattlesnake genome, and we're just looking at all the variation, and all the green branches are where we see that higher positive selection, so more change is happening. And I actually found significantly more changes happen in the extracellular part of these receptors. So a receptor, it occurs in the bound to the cell membrane, and you expect whatever selection is happening on this protein that's driven by the actual smell that it's smelling to happen on the extracellular part of that protein, because that's where the scent molecules bind to, right? The scent molecules aren't going inside of the cell. They're kind of sticking to the outside where that protein is exposed to, to the outside of the cell. So I thought that was a pretty cool finding. And this is the sort of stuff that we can do when we're measuring these sequences and looking at evolution, both on the scale of the organism, but also on the individual genes inside of the organism. And is that the last slide? Yeah, so that's the last slide. We wanted to give you guys some time to ask us questions, because I know we just dumped a whole bunch of stuff on you. But I don't know, did you have any last remarks?
No,
Who's got a question?
The overall thing I hope you're picking up is that what we're trying to do here, we're basically a genetics lab. We're trying to understand the genetics of these traits, but we're also linking that in with ecology, whether that's in feeding and how these things are able to feed or not feed on different prey species, as well as how they're able to sense them. And so, it's a sort of integration across genetics, ecology, and evolution, and how the genetics of traits might bias the way that evolution goes. And so this is sort of the repeating theme, despite having all of these different kinds of projects going on in the lab. Anything to add?
Well, maybe to that end, could you tell us about what you guys were studying before this? That was probably the coolest thing I could see, is the integration of the different fields, right? This is our kind of curriculum over here for AP Bio, and you touched on every single unit in cool ways, right? So, we brought you in for venom, right, and all different kind of species, which is cool, but you obviously got the component of genetics, you got ecology, you got cell signaling, which we just finished. So I had a question on that data table or the phylogeny you just pulled up.
Yep.
Is that based on the distinction or the differences between the proteins in the plasma membrane there, or is that on a species level? So what?
This is all
level
one
is that?
species. So
Okay.
there's a ton of copies of this type of gene in the genome, which I wasn't expecting. If I knew that, I would have chosen an easier project.
Yeah.
But
That's wild diversity.
and so I only demonstrated on this tree. So the positive selection is more light green, is more individual sites that are under positive selection, and this is only looking at the extracellular part of the OR receptor. So only that little piece that's on the outside, and those are all the changes that are happening just in that region.
Wow.
So,
Okay.
so yeah, you would predict that to have more of an effect on, binding to a certain scent or something, as opposed to the inside of the cell.
Okay.
Yeah. And the question of, as far as background and sort of how we get here,
Yeah.
I think this is actually an interesting thing to see how everyone that ends up as a graduate student in my lab typically has a very different background, and that's, I think, an important part of the research group as a whole, is that we're doing such a diversity of things that basically any skill set that comes in will have a new perspective on that. And so my background is actually mostly in computational biology. And so I didn't start working on venom until I've been at FSU for a few years. I actually studied viral evolution before that. So I was used to thinking about these types of things, but in terms of the evolution of viruses.
Yeah, and so my background is I was a snake guy. So I graduated high school, and I was like, I don't care what I do, I just want to research snakes. And so, I went to college, got my biology degree, and then I worked, I call it bio-bumming, where I just got seasonal positions on a bunch of different field projects where people were sampling, we need someone to go out in the desert and collect a bunch of rattlesnakes and get blood from them, and we'll pay you to do that. And I was like, that sounds like a perfect gig for me. As I started to get more involved in the field, I started to learn that things like venom, research were possible. And so that's kind of what led me to there. But
Yeah, so how I got into the venom world is I actually started college as an international business major, which was not what I wanted to do. So I got there because I didn't know that there were careers available in biology like this. And so I started thinking, I just want a career in business so I can make some money. But then I realized that when I was in high school and things like that, it was the animals and it was the biology that really drove my passion. I still loved going outside, hiking, camping, that type of thing. And when I got into college, I was like, okay, I can't stand another business class. I'm going to go to biology and so like just kind of start over from there. And so then I started doing biology and chemistry classes, and I actually come from more of a biochemistry background. So I did a lot of things with chemistry, and all of my research before this was in chemistry looking at different types of small molecules that fungus produced. And then I came here and I started doing all of the snake work. And so that was going out. Everyone in our lab goes out and we do field work. So we're out in the field, we're collecting the animals, we're seeing how they interact in the environment. We then bring some of those animals back and get the venom like we showed here. And then we do some of the computational stuff where we sit behind a computer for hours on end, typing into a black screen and see what happens. But yeah, that's kind of a transition for me that I had happen.
And the way I got into it, I was always, growing up, I was always really into animals and stuff, but I didn't really know what I wanted to do with that. And then in high school, when I took my version of AP Bio, I started getting really interested in genetics and evolution. So when I started my undergrad at Louisiana State, I reached out to a professor who was the curator of herpetology at their Natural History Museum, and I started working with him, and I ended up doing a small research project on blood proteins in reptiles. And so for that, I was just taking sequences that other people had already generated, and I was constructing phylogenies, so these like family trees, looking at the evolution of those and which groups they were present versus absent in. So I came into Darin's lab interested in gene family evolution and protein evolution, and then got really, really interested in the coevolution aspect and all the different interactions that are happening to drive the evolution of these traits.
So my question is, like with the genes that you guys collect, do you like make anti-venoms after to like help people who get bitten by any of these?
No, we don't do that part of it because we're not equipped for it, and that requires a whole nother setup and a whole set of approvals and things that we don't have, and we're not good at it. And so we let the people that are good at it do that. But our work has been used to improve that. So, for example, one of our earlier findings in venoms with the local species, the eastern diamondback rattlesnake, is that when you collect the animals down near Miami, they're missing a venom component that's super important and super abundant in the venoms up here. But all of the people producing venom used to make anti-venom were getting their animals in Miami. And so we published this, and they changed their collecting strategies so that everyone ensured that they were getting these northern population animals to get that toxin in there. And so even though we don't do it directly, everything we're doing, all of our results, all of our data ends up in public databases. It's available for people, and so it does get used for that. It's just not our stream.
Yeah, I guess just to add to that too, and Emilie made the same point too, like you can do these studies with sequencing information that's already available. In order for science to work, it has to be repeatable. Whatever your experiment is, whatever your complicated analysis with the D/NDS ratios, someone else needs to be able to go in there and repeat that and say, yep, I did the same test and I got the same result. The way we do that is all of our sequencing data, even the raw data before any filtering or anything, is all publicly available. And when we submit a paper for publication, all of those, they're called accession numbers, where you go into the database, and it's free. You guys can do this too. It's called the NCBI database. You can pull up a human genome. You can pull up our last rattlesnake genome that we had published. It's all available there for literally anyone with an internet connection. You could download a rattlesnake genome right now. It's about 1.5 gigabytes, and do whatever you want with it, and just take a look at it. So yeah, I think it's a good point. So that's we're telling people, here's what the venom is. Now it's your job to go find the anti-venom and make informed decisions based on that.
I had a question about the snakes. You said that you guys do a lot of field work and you guys go out and you catch them. Do you keep them? Do you release them? Do you use them for multiple studies? How does that work?
So we, all of the above, I guess. But for the animals that we do the research on directly, we have a collection at FSU that we maintain. And so we have our venom permit with the state. We have to get the university to come check our animals. There's a lot of regulation on the university level. It's called ACUC, but it's similar to like University PETA. They come in and make sure like whatever projects you're doing, you're not torturing the animals or anything like that. But before any of that happens, we have to put together, you know, a proposal and get state collecting permits and stuff like that. So yeah, so we do collect our own animals, but there are a lot of steps involved.
Do you guys have favorites?
Hey, y'all, let's give it up. Thanks so much.